In this video, we will learn about mitosis or mitotic cell division. Mitosis is one type of cell division by which one parental cell will produce two identical daughter cells. So if a parent has 46 chromosomes, daughters will also have equal number of chromosomes. So this is mitosis. So parents will give equal number of chromosomes to two daughter cells. So daughter cells have the same number of chromosomes as present in the parent. So this occurs both in plants and animal cell. So this type of cell division leads to growth of the individual and repairing and replacement of damaged part. Now, which cells will undergo mitosis? So all our body cells, which are known as somatic cells, will divide by mitosis. So what is the other type? The other type is known as germ cell or gametic cell or gametes. So gametic cell will divide by other type of cell division, that is meiosis. Except these gametes, that is male and female gamete, that is sperm, which is a male gamete, and egg cell or ovum. So except these two, all other body cells are termed as somatic cells and they divide by mitosis. Example, skin cell, liver cell, epithelial cell, etc. Now, mitosis can be divided into two phases or two sections. First phase is karyokinesis, also known as nuclear division. Now, karyokinesis again has subparts, that is prophase, metaphase, anaphase and telophase. Now, each step has its own importance. So here the main aim is to make one nucleus, so one nucleus of a cell to produce two nucleus. So nucleus has to divide step by step. So at the end, a cell should have two nucleus. So first is the prophase. Now prophase marks the beginning of the cell division. It is the longest phase of mitosis. During this phase, what are the changes that we see? One is the chromatin material will condense to form a distinct thread-like structures called as chromosome. So a typical chromosome will look like this. This is the actual photographed chromosome and a diagrammatic representation is on the right side. So here we can see in the center the most condensed area, that is the centromere or kinetochore, and to this attached are four arm-like structure. They are known as chromosomal arm. Now if I take these four arms and divide it into two, there is two identical hands. Those are known as chromatids. So since they are both attached to one centromere, they are termed as sister chromatids. Now each chromosome has two identical thread-like structures called chromatid and they are attached to only one centromere. Now centrosome is another structure which will divide in the prophase to form two daughter centrioles. And these centrioles will move to opposite poles of the cell. Now from these centrioles will be like this. These are microtubule, proteinaceous structure. So they will produce fine microtubules called astral rays which radiate out from each daughter centriole. So how will a cell actually look? So here we can see inside the nucleus the condensation of DNA is happening, and on the other hand outside we can see centriole has divided and produced two daughter centrioles and they have started to move to opposite direction in the second picture. So this is one centriole, this is the other, and as this moves to opposite sides there are fine threads which come out of it. These threads are known as spindle fibers. Now on the opposite direction we have shorter sections, these ones. These are known as aster rays. So here we can see asters. The centriole with its astral rays forms an aster. In plant cells they do not have this centriole. The centrioles and astral rays are absent. So if we draw a cell division phase in plants, so we should not draw centrioles or aster, but we have to definitely draw spindle fibers. So this is how early prophase and late prophase look. So as the late prophase comes near, the nucleolus will also disappear. The nuclear envelope, that is the membranes, will disappear, and we can see the distinct chromosomes with the centromere. Now we can say that microtubules between two asters form a spindle. The equator of spindle lies at the center of the cell. By the end of prophase, the nucleolus, nuclear membrane will disappear, while the chromosomes have become short and very thick, and it's easy to recognize or identify them under microscope. Now the next stage comes the metaphase. So metaphase, the very important phase, because the chromosomes are shortest and thickest in this phase, and they all lie under a straight line. So they line up on the equator of spindle with their centromeres on the equator and arms towards the poles of the spindle. So this is a metaphase plate, imaginary equatorial plane. So centromere lies in the middle and chromosomal arm, chromatid are towards the pole. Now spindle fibers are attached to centromere on one side, that is this part, and to the centriole on the other side. So we can see it is held together on both the sides so that it lies on the central plane. So this is how metaphase looks in case of an animal cell. So the left image is from an animal cell, because we can see centriole, aster rays, spindle fibers. But in a plant cell, what we should not write is the two structures that see here. Centrioles and aster rays are absent, but spindle fibers are present. So chromosomes are at equator of spindle, and we can see cell wall. Now comes the next phase, which is the anaphase, third phase in karyokinesis. So anaphase is marked by the migration of these chromosomes towards opposite poles. So how does this happen? The centromere, where the two chromatids are attached, that is these two. So it has to split in the middle. So centromere of the chromosome divides, and these two sister chromatids now form two daughter chromosomes, and they move towards the opposite poles. Now anaphase will also see a ring in the equator can be seen. That means it also starts to show the cell cytoplasm division. Now the contraction of spindle fibers pulls the daughter chromosomes away from the equator towards the opposite poles. So we can see half centromere and one of the chromatids, that is daughter chromatids, will be pulled towards one pole and opposite. So it should be equal number. So on both the ends we have equal number of chromosomes. Now chromosomes, since they are pulled apart, they can easily assume a shape such as V, that is the length of chromosomal arm are equal, V-shaped, or it can be a J-shaped, very short, or it can be L, little longer. So this shape will be assumed in anaphase. In anaphase of animal cell, we can see that contractile ring is formed at the equator. So it will begin to divide the cytoplasm as well. But in plant cell, parental cell in anaphase, there is no centriole or aster, and there will be no this contractile ring. And last phase in the karyokinesis is telophase. Now during telophase, chromosomes forms two groups, that is whichever chromosomal arm and chromatid was pulled away, they will start to localize on the opposite end in both animal and plant cell. So this either pole, the opposite pole reached chromosome will form their own group, and chromosome starts to uncoil and form chromatin net. So this is similar to what happened in prophase. So this is opposite of prophase. Spindle and aster rays disappear, and nuclear envelope and nucleolus start to reappear, and it can be seen as two separate daughter nucleus. And in the telophase, we can also begin to see the cytokinesis, which is the next stage. That is the division of cytoplasm followed by nuclear division, so that the parental cell is divided into two daughter cells. The process of cytokinesis is little different in plant cell and in animal cell. if we think of an animal cell, how does the cytokinesis proceed? So cytoplasm divides by constriction of the plasma membrane, which will begin on the equator and gradually deepens and coverages on all the sides. So it's like pinching off. So it begins on the sides and it will pinch off gradually. So this type of cytokinesis is known as cell furrow method. There is a furrow formed in the middle. And on the other hand, in a plant cell, there is a cell plate method where a cell plate is formed at the equator in the middle with the help of Golgi bodies, and this cell plate will extend towards the end. So cell plate is formed at the equator of spindle by fusion of vesicles that are produced by dictyosomes. Dictyosomes are simpler version of Golgi. Cell plate formation begins from the middle and proceeds towards the periphery. The primary walls are deposited on either side of the cell plate. So this will end the mitotic division in case of plant cell. I hope this topic is clear to you. Thank you.